All right, we are on page 177 of your binder. At this point, you have finished the chapter on meiosis. And if you need, I gotta skip all this. If you need help on anything with meiosis, email me questions. Make sure you have your questions ready because whatever I told you on the video for meiosis is exactly what I would have told you in class. So basically it was like I was there pretty much. So make sure you ask questions on that if you need to, because I am moving on. We gotta study that material, make sure you master that material because we are moving on to genetics. All right, so this guy right here, wearing this cute little dress and this apron. His name is Gregor Mendel, and he is considered the father of genetics. What he did was spent a lot of time breeding and studying peas, like the peas you eat. In case you didn't know, pea plants actually have flowers. There's purple flowers and there's white flowers. There are also different shapes and colors of peas, but we'll get to that later. I don't want to spoil the fun just yet. So why did he study peas in order to learn things about genetics? Because he can control the mating. Peas can't walk, so you wouldn't have one pea plant walk over to another pea plant and be like, hey, you want to do this? He could control the mating. He could do the cross-pollinating. There's many varieties of pea plants available, and I'm gonna show you what those varieties are. And then there's a short generation time, so he can collect his data after a really short amount of time. Some vocabulary words that you need to know from Mendel's pea breeding experiments. The pea generation is the first two subjects, so the first two plants that he used. And when we say they're true breeding, that means that their parents were, so this purple flower right here in the pea generation, it's both of its parents were purple. The white one, both of its parents were white. So basically, you guys know the terminology homozygous, heterozygous. A pure breeding plant would be homozygous for that trait. The F1 generation is the result of crossing the P generation. And then the F2 generation is the result of crossing the F1 generation. Okay, so pause if you need to. Just some vocabulary words. All right, these crazy looking things right here are chromosomes, and all this is showing you is that a gene for a particular trait that has alternate versions is called an allele. So the color for flowers on pea plants, there's a gene for that. They could be purple, they could be white. So it's the gene for color, but there is a purple allele and there is a white allele. Allele is almost like version of a gene. So just make sure you know that word. Ooh, we're just moving right along. All right, next page, 178. All right, so much information about pea plants that you just didn't know you're going to have the joy of learning today. Look at all of these different characteristics. I mean, oh my God, look, there's purple flowers and white flowers. There's axial buds and terminal buds. The seeds can be yellow or green, round or wrinkled. The pods can be inflated or constricted. The pods could even be green or yellow. And then the stems, the plant itself could be tall or it could be a little dwarf. So many different versions. What I need you to know from this chart, I don't need you to memorize what is dominant and what is recessive, but that you know the word dominant means it is the trait expressed, and the word recessive means it is the trait that's hidden. So if you have a dominant allele present, that allele will be expressed regardless of if the recessive allele is there also. And this is just showing you, look, on the left side are all the dominant traits, on the right side are all the recessive traits. So that's where we get our words dominant and recessive. So after Mendel spent all of his time breeding his plants, he found these four principles. Principle number one, alternate versions of genes are called alleles, and they cause variations in inherited characteristics among offspring. Cool. Principle number two, for each character or characteristic, every organism inherits one allele from each parent. And we know that from meiosis, right? One chromosome type, you have a chromosome type, one comes from the paternal side, one comes from the maternal side, so one from each parent. If two alleles are different, the dominant allele will be fully expressed. The recessive allele will have no noticeable effect on the offspring's appearance, unless both of the alleles are recessive. And then the law of segregation means the two alleles for each character separate during gamete formation. So law of segregation is basically stating that anaphase 2 of meiosis occurs. And this was before they knew anything about the steps of meiosis. So it's kind of cool that he was already saying this. So the next slide isn't really, I would just, basically that's just saying that the two alleles for each character separate during gamete formation. And it's just showing you a picture. So the two alleles separating during gamete formation and anaphase of meiosis gives us the ability to do Punnett squares to come up with probabilities of certain outcomes. All right, this is showing you dominant and recessive. So if you're using a dominant trait, it's going to have a capital letter. If you're talking about the recessive trait, it's going to have a lowercase letter. And then we have a couple more vocab words, homozygous and heterozygous. Homozygous means both letters are the same case. So either both letters are capital or both letters are lowercase. If it's heterozygous, it means you have one capital, one lowercase. All right. So what we're seeing here is that cross from before. I'm gonna go back. Wait, what did I do? Oh, this is what he did. He took a pure purple and pure white, crossed them, and got all purple. He took two of these F1 generation flowers, crossed them, got 75%, sorry, there's a really loud car outside, 75% purple, 25% white. So that's the F2 generation. So what we're looking at right here is the F2 generation. 3 were purple, 1 was white. But this is how, if you did the Punnett square, which I think, oh my God, I'm gonna give this a try. Oh Lord. Let me see if I can do this. Nope, can't do it. Pen color, black. Okay. Yeah, that isn't working, like at all. Yes, it is. What? Okay, I'm gonna do a Punnett square right here. So if he crossed the homozygous purple with the homozygous, and I know that clicking is annoying, but I can't help it, homozygous white, he would get all heterozygous in the F1 generation, right? So then what he did was took two heterozygotes, big P, little p. I don't think P is a good letter to use because sometimes it's hard to differentiate between the capital and lowercase. Those are two big P's. I'm doing this with my MacBook because I'm at home and I have no other choice because we need to move on. You see what I did there. All right, everyone should know at this point how to do a Punnett square if you took cell bio. But all I did was I put each one right there. That's a P, that's a big P. And then I just crossed them. So what I end up with, you see how this one is big P, that would be purple, and it's homozygous. Big P, little p, that would be purple, it's heterozygous. Little p, that would be white, it's homozygous recessive. All right, so hopefully that kind of jogs your memory a little bit. How do I erase this? Pen, eraser pen. I'm sorry, I'm not going to edit this because I'm going to edit these videos. It's too much work. All right, moving on. I guess that worked out. I mean, let's say we're all right. Okay, yeah, there we go. All right, couple more vocab words. Oh, just write this to the side. For some reason, this isn't on your notes page. Write it to the side. I just need you to know phenotype and genotype. Phenotype is the expressed physical trait, so you're gonna have to write that on the side. So you wrote homozygous, heterozygous. So right to the right of that, write phenotype and genotype. Phenotype is the expressed physical trait. Genotype is the actual genetic makeup. So phenotype is the what is the word I'm looking for? It's like the adjective. It's the description of what the thing looks like. Genotype is the letters. So that's how you can remember it. All right. We already did that one. I just did it for you a minute ago. I just did this one for you a minute ago. It was the one where, hold on, where you got this outcome right here. So I'm not gonna go through the torture of that again. We're gonna do some problems when I get back next week, so that'll be fine. I just wanted to make sure y'all had the notes on this and then I jogged your memory on it. All right, so let's say you're looking at a purple flower. Let's say you're looking at a purple flower. If you are looking at a purple pea plant flower, do you know whether it's big P or big P little p? You don't, because both of those options are purple. So how can you find out if it's big P or big P little p? You do this test cross, and you test cross it with a homozygous recessive. So if our purple flower was big P, and we crossed it with a white one, we get all purple flowers. And this Punnett square right here is showing you that. If our purple flower was big P little p and we crossed it with this homozygous recessive, you would end up with some white flowers. So all this is just a way to determine if the dominant trait is homozygous or heterozygous by crossing it with a homozygous recessive. So that's a test cross. Make sure you ask about that if you don't understand it. Oh my God, who's texting me? Okay. All right, law of independent assortment. This again, I don't like to get like, this is saying that the alleles are going to separate independently from each other during gamete formation. So once again, this is saying that sister chromatids are going to separate from each other during anaphase of meiosis. And that's how we can do Punnett squares. I think that's just as far as I'm going to go into it, because right here, it's showing you that in the picture. Here are two options for how they line up on the metaphase plate for meiosis. Here are the two options for metaphase 2, and then here are all the options for the gametes. It's just showing you the gametes are separating independently from each other. So the red ones and the blue ones, like, they could possibly line up different ways on the metaphase plate and then end up in different combinations. All right, and each combination is as likely as the other one. That's really all this picture is telling you. Okay. What we have done so far is what's called a monohybrid cross. A monohybrid cross is where you make 4 boxes. A dihybrid cross, ooh, people hate dihybrid crosses like so much. A dihybrid cross is where you're studying 2 characteristics at the same time and you're making one big old 16-box Punnett square. So just write down monohybrid cross, dihybrid cross, and just know that we will do some 16-box Punnett square practice next week. Let's write down a couple more things. Oh yeah, so okay. You know what? I'm gonna do the example so that you can see what this is. This freaks people out. All right, I want a black pen. Where is it? There it is. All right. So this is telling me that I have 2 traits. So let's say it's hair color and hair length. A is hair color, B is hair length. Brown is black is dominant to brown, long is dominant to short, something like that. Heterozygous, heterozygous. So what I would do, and I sure hope y'all learn this in cell bio, I would make a 16-box Punnett square. But guess what? Even though we're going to do practice doing 16 boxes, I'm going to show you the shortcuts. But in order to get to the shortcut, you have to go through the long cut. I don't even know if that's a thing. 16 boxes. All right. Up here, I am going to write down the different combinations of A's and B's that could possibly be in each gamete. So if you look at it, you do like this FOIL method. Big A and big B could be paired with each other. Big A and little b could be paired with each other. Look at that, look at that. Little a, big B, and little a, little b could be paired together. Cool. This is the exact same genotype right here. So I'm just going to write the same thing down the other side. But you're gonna want to write this down somewhere. I would say like on page 181 to the right of that one it's showing you, because they're not showing you this example right here. So go ahead and make sure you have that written down. All right, and then what do? I do the same thing that I did with my 4-box Punnett square. I combine them. I do capital letters first, and I keep same letters together. So you have big A, big B, big A, big B. I'm gonna write big A, big A, big B, big B. All right, so I'm gonna pause this, I think, so that you don't have to hear all the clicking, and then I'll put it back on when I'm done. Okay, so just kidding, it actually wouldn't let me do that. So now I'm, oh my God. Now I'm really confused on how to do this. This is so hard. Gosh, I just got this. I guess it's kind of working. All right. And my phone's ringing, so hold on. I have to go back to doing it the other way. I know this is annoying, but it is what it is. It won't let me stop the video and write it, which is fun. Little a, big B, big A, little a, big B, little b. Does everyone get what I'm doing? Like, if I just stopped right now, would you? No, I'm doing it. I'm going to the very end. I'm doing it. I mean, I'm almost half. I'm like halfway there. I might as well, right? Yes. I need someone to cheer me on. I can do it. I hope everyone gets why I'm putting the letters that I'm putting. Big B, little b. Maybe if I sing it, it'll be better. Big A, little a. Big B, little b. There we go. Oh my God, my arm is hurting. Big B. Yes. Yes. There it comes. And done. Okay. So the whole point of me doing this was the question will ask you how many or what proportion of the offspring are dominant for the first trait and recessive for the second trait. So what you would do is you'd go through this thing and you'd count them up. So I'm going to do a different color. All right, so dominant for both traits. Which ones are going to express dominant for both traits? It's because there's at least one big A and one big B. Oh, I skipped that one because there's no big B there. It's not dominant for both traits. All right, this one, last one, last one. No more. All right, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them are dominant for both traits. All right, what am I gonna pick? Red. How many are dominant for A but recessive for B? Dominant for A but recessive for B. And that's it. All right. Yellow. How many are dominant for B but recessive for A? Recessive for A but dominant for B. I hope you're doing this, and color coding it would actually help you out a lot. All right, pen color. Let's just do gray. And then recessive for both. Okay, so what you just witnessed here is if you are doing a dihybrid cross and both of the parents are heterozygous for both traits, you have this thing called the 9, 3, 3, 1 rule. 9 out of 16 are going to be dominant for both. 3 out of 16 are going to be dominant for the first, recessive for the second. 3 out of 16 are going to be recessive for the first trait, dominant for the second, and 1 out of 16 is going to be recessive for both traits. Okay, hopefully you have that written down, because that was a lot, and I don't want to have to go through that again whenever I already did it. So make sure you have that written down by the time you come to class. I'm erasing so that I can move forward. We're almost done. That is a dihybrid cross. I'm going to get you, we're going to do a little bit more practice, but you got the gist of it there. Okay, the problem is not every time are both parents going to be heterozygous for both traits, but there still can be other shortcuts available. We're going to do some practice next time I see you. All right. I'm done with you, Pin. All right, last thing. If we're doing probabilities, I'm going to show you another shortcut for those dihybrid crosses next time I see you, but I want you to write this down. Rule of multiplication means that there are 2 or more independent events that are going to occur together. You multiply them. And then, okay, so the key term is you multiply when you see the word and. So when you see the word, I'm all into this Pin now. When you see the word and in a word problem, you're going to use the rule of multiplication. All right. Kind of like, if I'm throwing a dice 2 times, what is the likelihood that I'll get 6s both times? You would do 1 out of 6 times 1 out of 6. Probability of having 5 boys in a row. 1/2 times 1, blah, 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 1 over 32. The rule of addition, the word you're looking for is, can someone tell me? You're right. You're looking for the word or. All right. So if you see the word or in a problem, you could actually add them together. So you add. Chances of throwing a die that will be a 4 or a 5. That would be 1 6 plus 1 6. All right. That will come into play when I show you some shortcuts and working some of these problems. Next time I see you, we're going to do non-Mendelian genetics. That would be codominance, incomplete dominance, blood typing, maybe a little pedigree practice, and then we have lots of fun problems to work on. I would like for you to do the 3 problems on page 186. Page 186, do 1, 2, and 3. Super easy, super easy. And then we will pick back up next time I see you. All right, bye.